Professor Barry Ninham AO FAA. Barry's discoveries have had a revolutionary impact on the field of colloid science, a discipline that underpins chemical engineering, cell and molecular biology and nanotechnology. He is the developer of the accepted theory of amphiphilic molecular self-assembly, a process that underlies modern material science. It is a fundamental principle of self-assembly in nanotechnology, impacting on modern molecular-based technologies and slow-release technology for in vivo pharmaceutical drug delivery. 5 decades of work by Barry Ninham has revealed that the discipline of physical chemistry that informed our intuition on a myriad of processes was flawed, to the extent that it failed to take account of key ion-specific effects and dissolved atmospheric gas. Barry, my congratulations. I invite you now to present the 2017 Matthew Flinders Lecture entitled The Physical and Biological Sciences Divide.
What happens? Nothing. Oh. The panel controls are locked. How do I unlock them? Oh. Okay. Thank you, Jim. Sorry about that. Got it. It was 100 years ago, exactly, since Darcy Thompson wrote a famous book called On Growth and Form. He was a polymath, and he made a point strongly that the founders of the cell theory of biology in the previous century and of the physiologists, they expected advances in biology to depend on progress in the physical sciences. It's 100 years on, and we are still divided, and I want to explore why we are divided and how far we've gone. The reality 100 years later is that we're still in a muddle, and I want to tell you what I think went wrong. We're at a stage where, although there's been huge advances in understanding molecular forces and self-assembly, that in dealing with biology, people in Harvard and other places are starting to get confused and to bring back the idea of vitalism. There's something magic about biology. There's something magic about the idea of enzymes. And the question is, where does the energy come from, and where does it come from with such excruciating precision? So we've got our own dark energy in physical chemistry, as the astronomers have, the cosmologists have in their area. So the question is why this is. There's a genuine confusion that we've buried, and it turns out that it's due to sins of omission. The Greeks told us that there were 4 elements. They were the earth, and salt, and water, and air, and fire, temperature. In our theories, we dealt with the earth, matter, and we dealt with water, sort of, but the others were missing. And those missing elements are the missing answer. That's where the dark energy is, as I'll try to explain. After we rebuild the theory, having recognised that, just very recently, we've been able to make some really astonishing advances. This is done in Richard Pashley's lab in ADFA, with whom I work. And the results are, I'm giving the end of my talk because I may run out of time. First, what's happened is that when we include these missing elements, then we've discovered some extremely effective new technologies to do high temperature reactions at low temperature. We've discovered ways to do desalination extraordinarily cheaply and easily. We've discovered how to deal with this recycled water, with viruses and antibiotics. And curiously there, the magic gas is carbon dioxide. And we've discovered how to clean up arsenic, lead, and radioactive waste and pollution, and to control cavitation in shipping that makes them go quieter. And so these are really amazing developments, and they're because we were missing something in our earlier theories. And climate changes can relax. These are really important things, and they can be done cheaply. The cost of this research is zero, by the way. Well, the reasons for the muddle where we got confused are, I think, a missimulation via computers. They were premature biological modelling by flawed theories from physical chemistry. It's not their fault. They had to do what they could do. There's primitive ideas of structure, the use of Euclidean geometries, spheres, cylinders, and planes, when the real structures are bicontinuous, and the forces were too crude. Now, these things affect things that were deeply embedded in biology, things like the Hodgkin-Huxley relations, ion pumps, anesthetics, lung surfactants. They're embedded very deeply, and they're hard to reconstruct. So let me deal with structure first. We're dealing with growth and form. Growth means forces, form means structure. So I'll deal with form first. And here's an example from years ago. Here's the structure of a real, nanostructure of a real microemulsion formed from oil and water and surfactants or lipids. The real structure is bicontinuous. It's interconnected tubes. You can do this structure with a conductivity meter and a piece of paper if you know volume fractions. We got confused on the way because in these recent years, people got onto big machines like neutron scattering from synchrotrons and spent an enormous amount of money. And the kind of structure of the thing I just showed you, they came up with for many years were just spheres of water in oil. Well, the structure is nonsense. The structure comes up because the program you write using the structure has spheres and cylinders. So this was a diversion. Here's the real structure from recent electron microscopy. You see it's bicontinuous tubes, not spheres. Here's another structure, lung surfactants, which we did recently. I won't go into details. It's rather complex, opening and closing, with the holes have got special proteins there. But that's the actual structure, but the structure that people working with lungs in the medical field were dealing with had envisaged a monolayer of phospholipids on top of a multi-layer of other lipids called tubular myelin. The structure they dealt with was complete nonsense. It's not their fault. It couldn't possibly work. But when you get the right structure, then you can do some real work. On forces, on structure, we're not so bad because people have now understood about bicontinuous cubic phases. My colleague Stephen Hyde and others have made enormous advances there. But on forces, we're in a bit more of a trouble. Well, let me tell you the scene. In 1986, I was at a Nobel symposium, and it was dealing with protein structure. And the lead speaker was a Swiss-German guy who was very confident, and he stood up and he had a slide that said NMR. NMR, nuclear magnetic resonance, obviously. No. He said, no. NMR means no more research. The problem solved. The science is settled. Now drug design is easy. Why? Because we can now simulate on a computer with 62,000 molecular parameters any protein or enzyme we want. Marvellous. There's no water in this simulation, but at the end of his lecture, an English quiet biochemist said, well, what about my enzyme? It denatures at 37 degrees. Ah, easy, said the German. We just changed the parameters. The science is settled. Well, that kind of research has got bigger and bigger and bigger since. It's a large part of the NIH budget, and people buy these programs and they simulate these proteins and change the parameters. So the problem, our dark energy is still there. The problem is enzymes. Where does the energy come from? Well, free energy runs downhill. So what? That doesn't mean anything. There's a problem with ion pumps. There's dogma there due to flawed theory. And lots of other areas. So we've still got this problem. We've got, well, other problems too that I'll skip over. One of the difficulties we had in theory, which are now solved, dealt with something called Hofmeister effects, specific ion effects. Specific ion effects means it's a mnemonic to say that we don't have a theory. The theories that were used in biology, in Hodgkin-Huxley equations and things like that, ignore specific ion effects. They've been around for a long time and we haven't been able to deal with them. That problem has now been solved. And let me show you how it turns up. If I do a very simple experiment, I'll measure the pH of a salt solution. I've got a sodium phosphate buffer. I have a theory of this. That's standard, classic, 150-year-old physical chemistry. So we measure the pH as a function, pH here as a function of salt concentration. I've got a buffer, so I've got rid of carbon dioxide. And these different squares and triangles measure the pH as a function of concentration. For sodium salts of different anions, bromide, chloride, acetate, and so on. And you see they're all different, and the difference is quite significant. It's a considerable, it's more than a pH unit. So the theory doesn't accommodate that at all. And the measurements don't make any sense. If I go further and do more of these simple experiments with pH and with buffers that we take as given, this Hofmeister sequence, the way the different anions change, or cations change with different salts, it reverses if I, the sequence reverses if I change the cation from sodium to potassium. It shouldn't. It changes if I change the buffer, it reverses from one form to another. If I change from phosphate to another one at the same nominal pH, surface potentials change in sign. Reverses from sodium to potassium, and it's impossible to make sense of these simple measurements in terms of classical electrostatic theories. And yet our poor friends, the biologists, have been taken in by this notion that this stuff means something. It doesn't, it's nonsense. So and we now know why and can fix it up. Here's my archetypal restriction enzyme problem. I've got an enzyme here. It's got a hydrophobic pocket. It's got an active site to which I've got to bind a specific divalent ion, magnesium, whatever's required. And I've got linear DNA. This is a cartoon of a restriction enzyme called Hindi-2, which is a classic tool that you use in molecular biology, and we wouldn't be without them. They're good for chopping up DNA into bits in a precise way. Well, if we move out of the standard background and do my specific ion effects and study the restriction enzyme cutting efficiency, I'm sorry about this slide, that's supposed to be the percentage of cutting of linear DNA up here, and as a function of the cation, that's the positive charge concentration as a function of added salt. And I'm looking at the efficiency for a particular salt, a bunch of salts. The different colours represent different salts, different cations and different anions. So the efficiency goes up, say for the purple one, it goes up to a concentration of 0.15 molar, and then it goes down and tails off. So they're all different. There's nothing in, one has a series of results for different salts. The other has a series of results for a different buffer. I've got the same buffer. Classical theory says I should have the same results. I've got the same nominal pH. It reverses for different buffers. So this thing doesn't make any sense at all. So we're getting ourselves into a muddle. We've missed out on temperature. We've missed out on Hofmeister effects. We missed out on quantum mechanical forces, and that area we did an enormous amount in my own department and did lots of work on measuring these forces and to working out how to include them in the theory. We missed out on something that I'll come to called dissolved gas in the atmosphere and so on. So we were going along in this confused state in terms of relating to biology and to other things too, chemical engineering and colloid science. And then suddenly, a few years ago, 20 years ago, we realized that even though we'd done all the fancy quantum mechanical forces, quantum electrodynamics at the highest level, and sorted it all out, we'd made a fundamental mistake in the theories of physical chemistry. Ignoring hydrophobic interactions, dissolved gas, we'd made a fundamental mistake in the beginning ansatz of physical theory. We'd assumed that we could treat electrostatic forces, double-layer forces, by a nonlinear theory, and the quantum mechanical forces, which are highly specific, by a linear theory. Well, that violates some fundamental principles of physics. So we were wrong at equation 1 in this 150-year-old science. So we had to fix it. So we fixed that. But then there was even more confusion because there's something called long-range hydrophobic forces that biologists have known about for a long time, and it's part of their atmosphere. Then we found that hydrophobic forces disappear if we remove dissolved atmospheric gas. Now, people don't believe that because the dissolved atmospheric gas is very low. It's 10 to the minus 3 molar oxygen. Why the hell should that make any difference? Well, it does, in the same way that defects in a solid make a huge difference. A lot of strange things happen when we, and I've listed them here, and I won't dwell on them because I'll come back in the end. So things change if you remove the dissolved gas. This is where our dark energy comes into it. As I said, we did an enormous amount of work in my department from 1970 on exploring all these forces, which happened to be relevant to particle physics too. The fanciest physics you could possibly imagine. We produced 100 professors working on this stuff, and they all did wonderful things and went off. But we had missed out on the dissolved gas, and was something, even though we fixed the theory, something was wrong. And the startling thing happened 30 years ago, and people have ignored it, except for Richard Pashley, myself, and another colleague called Vincent Craig. I did this experiment 30 years ago when I opened up an international conference we had here involving 2 international unions. And the experiment is this. Consider a fish tank. We've got a bubble, a column, and I've got a frit, like a fish tank, with bubble air through it. And I'll bubble the air through, and if you look at it, you see that the bubbles come out of the frit and they collide, and they get bigger, and the column stays clear. The bubbles get bigger and bigger. But then I add some salt. You can do this with a glass of beer too. Add some salt, and all of a sudden, at 0.175 molar, exactly the concentration of salt in the blood of us and reptiles and every other animal that came slithering out of the Permian Ocean a while ago, and the Cambrian Ocean too. All of a sudden, the bubbles won't fuse. The column goes whoosh. It's a fantastic experiment for first-year students. And they won't fuse. And here's the percent coalescence as a function of salt. All of a sudden, very sharply, at physiological concentration, they won't fuse. So that happens for all salts, except for all the other salts. There are one group of salts that do this, and another group of salts that don't. There's no effect even up to 6 molar. So what the hell is going on? This has got nothing. There's a set of rules that emerge. There are a set of rules for different mixed salts. It's like the periodic table. No one, after 30 years, has the slightest remote understanding of why this occurs. So if we can't consider the ocean and answer the question of why it's foamy, and yet fresh water is not, and why fish tanks behave the way they are, we seem to be in big trouble. And the big trouble is that we missed out on the dissolved gas. We know that, and we can show that if we go further and we take notice of the Greeks, we ask, okay, we've got earth, we've got salt and water. Now let's add temperature. We've got air in the bubbles too, in dissolved gas. We add temperature, we cook up the column. we cook up the air. So we take our frit and we heat up the gas. It's easy to heat up the gas, it's cheap, much cheaper than water. So we heat up the gas and see what happens. Well, what happens then is if we start to do reactions in our column, the gas doesn't heat up the water stays at a fixed temperature. But if we keep on passing through hot water, we can do high temperature reactions, very high temperature reactions at very low temperature. So there's something about the surface of the bubbles of the water-air interface which is highly catalytic. So we can do these high temperature reactions. And then, as I said, Richard Pashley has developed techniques to do desalination that are very, very cheap, involving exactly this technology. We can use, if we use carbon dioxide in our column, we can kill off viruses very, very effectively. We can use air too, but carbon dioxide is much, much more effective gas. And as I said, we have ways of cleaning up arsenic, mercury, lead, and radioactive waste. And we can also do something else that I'll show you. If we remove the dissolved gas, which I told you is very small, you can stop cavitation. This has been known for 200 years. If you try to pull water apart, cavitate, open it up, it takes 200 times less energy than theory would tell you from molecular forces. And so if we remove the dissolved gas, you can't break up the water, and so you stop cavitation, and ships will go quiet and faster, and you can sneak up on your Chinese people with your submarine and do a good job. There's a whole new revolution there available with cavitation because we can make ships go much faster. And here's an example. It's not a really good slide, but if you see that propeller there, it's whizzing around, and you can see like an outboard motor on a propeller. You get this very large noise when you go too fast and you're breaking up the water and it goes white, and there's the cavitation there. But if I do a trick and I project gas-free water onto the blades, you can see the blade is perfectly clear and the thing goes along very quietly. The major source of energy for driving ships is due to this cavitation effect. So we can remove it, which is neat. Desalination, I don't know how I'm going for time, Jim. How much?
I don't know how you're going for time. There's supposed to be a little timer that comes up in 5 minutes.
Oh, well. Oh, is there? Oh, so I've got 5 minutes. Okay. Okay, well then I can declaim a bit more. Desalination is really neat. We use ion exchange, which was used by Ulysses in his voyages and described by Homer and Aristotle. And it's a clever trick that I won't get into because it's too long, but it's nice that they knew about these things a bit. So let me just summarise. As far as forces go in biology, and what I'm talking about also is true for chemical engineering, for disposal of nuclear waste, for a whole lot of fields that my department's worked in. It's all moved on. There are changing concepts and complexity with dissolved gas that we've ignored, with ion specificity that we've got a handle on. And these require a complete revision of physical and electrochemistry. And they have implications for things like ion pumps, conduction of the nervous impulse, anesthesia, and so on. And they open up a whole new territory. So we think that, oh, I forgot. There's any explanation for our dark energy for enzyme kinetics? Yes, there is. That's the whole point of it. There are these, I told you about the gas. Here's my problem. And what happens is this, it seems. The enzyme binds to the DNA and it dimerizes and moves up and down the DNA. We know all that. It diffuses to a hydrophobic nucleotide sequence. That's stereochemistry. Then you get this hydrophobic cavitation in the active site. And that produces energetic radicals, which is known from solar chemistry. And these give the energy to cut the thing. We've done a lot of work on that. And this is one enzyme, not all. But it's a method you can see by which nature harnesses all the weak physical chemical forces cooperatively to produce the chemical engineering. And it does it quite well because we exist. There are implications for this too, we can guess for contingency in evolution. The Ediacara extinction 50 million years ago, or the Burgess Shales extinction that Stephen Jay Gould writes about. He says it's contingency. We don't think so. We think it's because of after the Ice Age, the water, the salt temperature drops, the concentration drops below 0.15 molar, and that induces cavitation inside the single-celled animals, and they all died of the bends. So they all coincide with known carbon dioxide cycles. So this is a simpler idea of why we have evolutionary extinctions, but whether it's right or not doesn't matter. Acknowledgements, I'd like to say we were very fortunate. I was very fortunate in my time. We produced over 100 professors in my department who were, over the years, somehow, 5 of them became chairs of the Nobel Prize Committee in Chemistry. We had a lot of fun, we did a lot of work, and we made a lot of progress. And these particular people, Stephan Marcellia, Stephen Howard, Vince Craig, Rick Pashley, and my friend in Florence, where I work, have contributed to all this stuff. Well, that's about all I'll have to say. Thank you very much for listening to me.
Thank you, Barry, for that very stimulating lecture. Now we'll take some questions. If you put up your hand, the microphone will be brought to you. And if you wouldn't mind saying your name, then we can move on. Phil here and then Tony.
Thanks. A question out of ignorance, Barry. Cavitation and coalescence of gas gas bubbles, that's what it is?
No, they're different. They're different phenomena. We simply, I'm ignorant, been 30, 50 years in the game. Nobody knows why bubbles don't fuse above 0.175 molar. We simply don't know. So we can exploit that, and we are exploiting it, but we don't know. Despite all the fanciest theory in the world, we don't know the answer to the simplest possible experiment. And I use that as an example that the science is not settled. We hear that often. We don't know the answer to the simplest possible problem. Cavitation it's due to a different phenomenon. We've got a small amount of dissolved gas in water. It's about 10 to the minus 3 molar from the atmosphere. And that means there's one gas molecule about every 30 angstroms apart. If you think of the water as a solid, if it was a solid, the strength of the solid depends on defects in the solid, molecular defects. And the strength of a solid has got nothing to do with the strength of that material that you calculate from solid-state physics. It's due to the defects. In the water, these atomic dissolved molecules of gas affect the strength of the material. And that changes it by a factor of 200. So if you remove those dissolved gas particles, whether they're oxygen or carbon dioxide or methane, the water becomes enormously strong. It's hard to break it up.
Thank you.
Thank you, Phil. Tony?
Tony Klein, University of Melbourne. I noticed on one of your slides the proposition of high temperature reactions at low temperature. Now, I don't know if this has anything to do with it, but collapsing bubbles generate enormously high temperatures when the surface tension energy converts into thermal energy. Is this got anything to do with the price of fish?
Yeah, absolutely. Franz Grieser in Melbourne has done a lot of work on solar chemistry. It's really fine work, and it's directly relevant. Yeah, we don't understand the theory of this very well at all, but certainly that's the source of the energy. Hydrophobic cavitation is produced by a cooperative action of weak intermolecular forces, and then once you've got that small bubble there, you produce the, because of the high temperature, you produce free radicals. Yeah.
We can take one more question. No? So I think that's the end of the questions. Well, thank you, Barry, for that very, very stimulating presentation, very thought-provoking.